The pharmacokinetic barrier. Thermodynamics of drug absorption.
Biological membranes, and eventually into the bloodstream. Traversing that lipid barrier is a complex interaction governed by strict thermodynamic and structural rules. This infographic maps out the core framework of that process, detailing the 5 distinct mechanisms biological systems use to manage absorption. The specific pathway a molecule takes is dictated entirely by its physicochemical profile, specifically its lipid solubility, its ionization state, and its physical size. Looming over all these factors is the overriding force of the concentration gradient, which serves as the primary thermodynamic driver for molecular movement. The biological membrane acts as a highly selective active filter. Understanding the physics of these 5 mechanisms is the only reliable way to predict whether a given drug will actually reach its target. Looking closely at the first column of the diagram, we see passive diffusion. This is the default and most common mechanism for drug absorption. Notice that this process requires 0 cellular energy. It is driven strictly down a concentration gradient, moving molecules from areas of high concentration to low. The rate of this movement is dictated by a mathematical absolute, Fick's law of diffusion. Based on this equation, the absorption rate is directly proportional to 2 primary variables, the available surface area of the membrane and the steepness of the concentration gradient itself. The drug's lipid solubility, or partition coefficient, also heavily influences this rate. The more lipophilic a drug is, the faster it dissolves into and diffuses through the lipid bilayer. Because it relies purely on physics, passive diffusion offers a critical clinical advantage. It is non-saturable. As long as the concentration increases, the rate of absorption increases proportionally, without an upper limit. Dropping to the bottom left corner of the chart, we find the major modulating variables for passive diffusion: environmental pH and the drug's pKa. These dictate the ionization state of the molecule. The strict rule of the lipid bilayer is that only the unionized, uncharged form of a drug can successfully cross. Clinically, this means weakly acidic drugs absorb most efficiently in acidic environments, like the stomach, where they remain unionized. Conversely, weakly basic drugs require the alkaline environment of the intestines. Passive diffusion is a highly efficient thermodynamic process. It offers limitless capacity, provided the drug's chemical structure and the surrounding pH align perfectly to allow entry. That raises an immediate physiological problem. How do highly polar, hydrophilic, or water-soluble molecules bypass a barrier made strictly of lipids? Column 4 illustrates the simplest bypass: filtration, or aqueous porous diffusion. Instead of simple concentration, filtration is driven by hydrostatic or osmotic pressure gradients, pushing water and solutes between cells. But water-filled pores impose strict physical limitations. Molecules must be exceptionally small, typically under 200 daltons, and they must successfully navigate any opposing electrical charges lining the pore. For larger polar molecules that cannot fit through these pores, like glucose or amino acids, the system relies on the mechanism shown in column 2, facilitated diffusion. This still requires no ATP, as molecules continue moving down their concentration gradient. However, the workload now shifts entirely onto structural transmembrane carrier proteins. These proteins operate on the principle of specificity. They utilize highly specialized binding domains that only accommodate molecules with a specific geometric and electronic fit. This graph illustrates the critical pharmacokinetic limitation of that structural reliance. Because there are a finite number of carrier proteins, the process is saturable. As concentration rises, the absorption rate eventually hits a hard ceiling, known as Vmax. Furthermore, when structurally similar molecules are present, they compete for access to those limited transporters, which can significantly slow overall absorption. By utilizing cellular infrastructure, the body can import difficult molecules, but that infrastructure introduces severe bottlenecks, rendering absorption finite and highly vulnerable to interference. This brings us to a second major challenge. How does a biological system absorb essential nutrients or drugs against a steep concentration gradient? The third column demonstrates active transport. Its defining characteristic is a strict dependence on cellular energy to force molecules from an area of low concentration to high. This divides into 2 forms: primary active transport, which directly consumes ATP, and secondary active transport, which harvests energy by writing coupled ion gradients. Because active transport also uses structural carriers, it shares the exact same vulnerabilities as facilitated diffusion. It requires specificity, it can be saturated, and competitive inhibition. But active transport also has a defensive application. Cells use these same energy-dependent mechanisms for outward transport. Efflux pumps, like P-glycoprotein, act as security systems. When a drug enters, these pumps capture it and mechanically force it back into the extracellular environment. A biological system actively fights pharmacological intervention. Achieving absorption means overcoming kinetic cellular forces that are burning energy to pump the drug out. We are left with one extreme edge case. How exceptionally massive therapeutics, like large proteins, vaccines, or nanoparticles, breach a membrane when they completely dwarf standard transporters? The final column illustrates the solution: endocytosis, or vesicular transport. Rather than passing a molecule through the barrier, the cell expends a massive amount of energy to physically deform its own membrane, wrapping around the target to swallow it whole. This vesicular transclearance appears to be a nonspecific intake of extracellular fluid, known as pinocytosis, or it can be highly targeted through receptor-mediated endocytosis. When molecular physics and transporter mechanics are insufficient due to sheer mass, the cell must resort to physically engulfing the therapeutic agent. Zooming out from the behavior of individual molecules, we can see that membrane traversal functions as a strict, predictable biological rule set. This summary comparison table clearly divides the process. The thermodynamic drivers, passive diffusion and filtration, rely entirely on unionized states, physical size, and pressure gradients. Contrast this with facilitated and active transport, which demand precise protein interactions. This structural reliance introduces an inescapable limit of saturability, capping absorption speed. Meanwhile, these energy-dependent pumps can reduce a drug's efficacy by pushing it out, demanding precise dosage. Rational drug design requires precise engineering of a molecule's pKa, lipid solubility, and transporter affinity. Identifying a target receptor is just the very first hurdle in ensuring a drug actually enters the biological system. Administering the right drug in the exact right amount achieves nothing if the molecule cannot survive the biological membrane. Mapping these 5 mechanisms is the uncompromising first step in drug action.